Let's jump right in. Welcome to this explainer. We're going to explore the absolutely fascinating, dynamic boundary that makes literally all cellular life possible. We're talking about the cell membrane, and specifically, that incredibly elegant framework scientists use to make sense of it all, the fluid mosaic model.
You know, every single cell faces this fundamental biological imperative. It has to shield its highly organized internal world from the relentless chaos, the entropy of the outside world. It needs a highly selective barrier to maintain internal homeostasis, all while dynamically managing the constant flow of ions, nutrients, and waste. And this brings us to an incredible concept that essentially makes complex life possible.
That concept is compartmentalization. It's all about dividing the cell into distinct, managed zones to execute different functions efficiently. This crucial separation is what allows cells to maintain a perfectly stable internal environment, completely isolated from whatever is going on outside. It's what separates a tiny organelle from the cytoplasm, and ultimately the cell itself from the rest of the world.
Okay, so let's dive into this by assembling the cell membrane piece by piece. We'll start with the concept, then move to the phospholipid bilayer, cholesterol, proteins, and finally how this entire model has evolved.
Part 1. The concept. The blueprint of the cell boundary.
To really grasp how this boundary functions, we have to look back to the foundational blueprint proposed in 1972. S.J. Singer and Garth L. Nicholson brilliantly described the membrane as a fluid, dynamic mosaic of alternating globular proteins and lipids. And this was completely revolutionary at the time. Why? Because they didn't see the membrane as this static, rigid wall. Instead, they envisioned a two-dimensional, liquid-like solution. It's fluid because the lipid molecules are constantly moving around, giving the membrane its vital flexibility, and it's a mosaic because it's studded with all these various proteins of different shapes and sizes.
Moving on to Part 2, the phospholipid bilayer. Think of this as the fluid lake of the membrane.
So, these amphipathic phospholipid molecules are characterized by their dual thermodynamic properties. Basically, they have a split personality. Their hydrophilic, or water-loving heads, seek out interaction with the watery environments of the cytoplasm and the extracellular matrix. Meanwhile, their hydrophobic, water-fearing, tails cluster together in the middle to completely avoid that water. Because of this, they spontaneously drive the formation of a highly stable bilayer. This structural arrangement inherently blocks most water-soluble molecules, making sure only specific lipid-soluble substances, or actively transported molecules, can pass through that hydrophobic core.
Part 3. Cholesterol, the temperature regulator.
Now, if phospholipids are the fluid lake, cholesterol is the essential molecule that helps it adapt to shifting environmental conditions. It physically interacts with those lipids to stabilize the whole membrane. Picture this. At high temperatures, when phospholipids have tons of kinetic energy and want to rapidly drift apart, cholesterol physically pulls them closer together. This reduces fluidity so the membrane doesn't just fall apart and lose integrity. But on the flip side, at low temperatures, when those same lipids want to freeze and pack super tightly together, cholesterol literally gets in the way. It prevents them from packing too densely, which actually increases fluidity. Its rigid steroid ring acts as this absolutely perfect thermal buffer, no matter the environment.
Part 4. Proteins, the mosaic tiles.
Floating within this stabilized lipid structure is the membrane's specialized functional machinery, the proteins. First up, we have intrinsic or integral proteins. These are amphipathic molecules that literally span the entire width of the bilayer. Then we have extrinsic or peripheral proteins that just attach to the surface without crossing through. And finally, we've got glycoproteins and glycolipids, which form these intricate, complex carbohydrate structures on the outer edge of the cell. Together, all these pieces are actively transporting materials, facilitating super rapid signal transduction, and providing those absolutely vital cellular recognition mechanisms. They're the gates and guards of the cell.
Finally, Part 5, the evolution, a dynamic, evolving model.
You see, while that 1972 blueprint was revolutionary and gave us an incredible foundation, our understanding of this microscopic world has dramatically transformed over the decades. We now know the membrane isn't just some free-flowing empty sea. Fast forward to the 1984 mattress model, which showed that lipids actually actively adapt to the physical size of proteins. Then, in the 90s, scientists discovered lipid rafts, these specialized thicker domains of lipids that move together as a unit. By 2013, the model was officially updated to include physical restraints from the cell's underlying skeleton. And now into the 2020s,
Thermodynamics research has introduced the excitable fluid mosaic. It's a highly regulated, crowded landscape that is actively evolving. Which brings us to a really fantastic quote from Garth L. Nicholson in his 2013 update of the model. He stated that, quote, cells can be considered completely integrated mechanostructures. What he means is, instead of just floating around randomly, proteins and lipids are highly organized into specialized domains and functional rafts. They're structurally integrated right into the cell's internal scaffolding and the external matrix. It's all connected.
When we look at the modern model, what really stands out is the sheer density and crowding compared to that sparse 1972 version we started with. Proteins and lipids cluster together tightly into distinct domains. And this crowding is super important because it limits random movement, keeping specific enzymes and receptors parked right next to each other so they can signal and function incredibly efficiently. It's like a bustling city center rather than an open ocean.
And then there's the cell's underlying skeleton, the cytoskeleton. It actually corrals those proteins into specific confined zones in what scientists call the fences and pickets model. So the proteins might technically be sitting in a fluid, but they're largely confined to their specific neighborhood block by these physical fences. This makes sure they stay exactly where the cell actually needs them to be.
But wait, the story gets even more fascinating. Recent research highlights this cutting-edge concept of the excitable membrane. For the longest time, we thought lipids were just a passive solvent, just water for the protein fish to swim in. But it turns out, lipids actually act as an excitable matrix. They actively respond to changes in temperature, tension, and pH to physically shift the cell's state. These phase shifts actually control protein sorting and can even generate propagating pulses, exactly like we see in nerve signaling. The lipids themselves are incredibly active participants in the whole shebang.
So we're left with a pretty profound realization. How does this incredibly complex, constantly shifting microscopic mosaic manage to perfectly orchestrate every function of life? Next time you think about biology, take a second to remember this beautifully dynamic, crowded boundary. Picture those active lipids, that stabilizing cholesterol, and the corralling cytoskeleton, and ask yourself, what other microscopic mysteries are just waiting to be uncovered right beneath the surface of the cell? Thanks so much for joining me for this explainer. I hope it gave you a brilliant new perspective on the fundamental building blocks of life.